Okay, welcome to this next video in which we are discussing the adrenoreceptors. So we're still looking at the alpha-1 adrenergic receptor, and we want to see how its stimulation is going to lead to the contraction of smooth muscle cells. And we know that in order to begin the contraction of smooth muscle cells, what we need to do is phosphorylate the regulatory myosin light chain. Okay, right. So we've discussed that the activation of the alpha-1 receptor leads to the production of IP3, and then IP3 is going to bind to IP3 receptors and trigger a conformational change which takes these IP3 receptors from being in a state where they will be inhibited by calcium to being in a state where they will be stimulated to open by calcium. Okay, right. So I now need to discuss with you two different types of signaling that will occur through this. Okay, so overall what's going to happen is we're going to release calcium, but you can get different types of calcium signal being produced depending on how many alpha-1 receptors you actually stimulate on the cell. Okay, so let's talk about the two different forms of signal that we're going to get. So, one is a local signal, and the other is a global signal, okay? And basically, the local signal is going to occur when you get local activation of alpha-1 receptors on the smooth muscle cell. So, okay, let's say that the alpha-1 receptors will be stimulated at a certain portion of the smooth muscle cell. So let's say here, this pink portion of membrane that I've outlined, you're going to stimulate the alpha-1 receptors there, but not all over the cell. Whereas in the global signal, you will stimulate the alpha-1 receptors all over the cell membrane. And the type of calcium signal that then this leads to is going to be quite different, basically. Okay, so we'll start off with what occurs in the local signal. Okay, so usually what will happen is you will give some agonist for the alpha-1 receptors, maybe adrenaline or noradrenaline, most likely noradrenaline, and it will be a short-term signal, so you'll get some noradrenaline, it'll activate the alpha-1 receptors for a little bit of time, and then the noradrenaline will go, okay? And whilst the noradrenaline was there, it will have activated the alpha-1 receptors to activate the phosphoinositide pathway, and you'll have produced a little bit of IP3 in the region of those receptors. Okay, so IP3 will go up in the cytoplasm around where those alpha-1 receptors were activated. And this is going to cause a calcium signal from the ER that is called a calcium puff, basically. Okay, and I'll explain how it's going to occur. So if we now look at the ER, okay, let's draw a little picture of the ER. Basically, the ER is going to have IP3 receptors all over the place. Okay, so I'll draw some of these like so. So here's an IP3 receptor, here is an IP3 receptor, here's an IP3 receptor, here's another IP3 receptor, okay, and maybe just one more here. Okay, now, let's say IP3 has gone up in this area here, okay? So IP3 went up in this sort of area, okay, like so. And all of the IP3 receptors in this area have therefore got IP3 bound to them, okay? So then what needs to happen is one of them needs to get calcium binding to those 4 calcium stimulatory binding sites for it to actually open. Okay, and this is a part where there is a bit of a mystery. We don't yet know how this occurs. We don't know whether what happens is just one of them happens to bind 4 calcium ions from the cytoplasm, because of course there are calcium ions in the cytoplasm. They're extremely low and rare basically, but it could happen in principle, but we think what's much more likely is that you will also open some calcium channels that are in the plasma membrane, basically. Okay, so if this is the plasma membrane, you might also open a calcium channel in the plasma membrane, and we'll come back to this later. We think that you might also open a calcium channel in the plasma membrane, which will let in some calcium from the extracellular fluid into the cytoplasm. And this calcium that comes in from the extracellular fluid into the cytoplasm, that will then activate one of these IP3 receptors. Okay, so it will get 4 calciums bound to its stimulatory calcium binding sites, and it will then open, releasing calcium from the ER lumen into the cytoplasm. This will then spill over onto its neighbours. Okay, so its neighbours are this one and this one. Now for this one's got IP3 bound to it because it's in the area where IP3 levels went up. So it has stimulatory calcium binding sites available. So calcium will bind to those and activate the opening of that receptor. So it will then release calcium from the ER. Then the calcium will spill onto its neighbour, and it again has IP3 bound to it, so it will be activated to open. Calcium will spill onto this one, and this one doesn't have IP3 bound to it, and it's the same scenario for this one over here. These ones around the point where the IP3 went up, these don't have IP3 bound to them. So when calcium spills out over onto these IP3 receptors, what will happen? Well, calcium will now bind to the inhibitory calcium binding sites and will inhibit them even further, so they definitely will not open. Okay, so you will get calcium being released from the IP3 receptors that are in the area where the IP3 went up. Okay, and that will cause a local calcium signal in the cytoplasm where the IP3 went up, basically, and that local calcium signal is then called a calcium puff. Okay, so you get a local calcium signal within the cytoplasm. So a local stimulation of the alpha-1 adrenoreceptors on the surface of the smooth muscle cell membrane would then produce a local signal of calcium in the cytoplasm of the smooth muscle cell. Okay, so that's what's known as a calcium puff. Now the important other thing to add here is that basically once calcium gets to a certain level in the cytoplasm, okay, i.e. once the IP3 receptors have released a certain amount of calcium and the calcium has got to a certain height within the cytoplasm around the IP3 receptors, it goes from having a stimulatory effect, i.e. stimulating the opening of these IP3 receptors which have got IP3 bound to them, to having an inhibitory effect. Okay, so once the calcium gets to a certain height, it will actually inhibit the IP3 receptors and they will close, and then you'll stop releasing any further calcium. Okay, and then, of course, once the alpha-1 receptors are no longer being stimulated, then you'll stop the production of IP3. The IP3 will be broken down, so you'll lose the IP3 from the receptors, and meanwhile, you'll also have a pump in the ER membrane, and I can only show it down here because there's no space over there. Okay, and this pump is known as the sarco/endoplasmic reticulum. Okay, so the sarco/endoplasmic reticulum calcium ATPase. Okay, and that should be a / there. Sarco/endoplasmic reticulum, and then calcium ATPase. And for short, the sarco/endoplasmic reticulum calcium ATPase is abbreviated to SERCA. So S for sarco, E for endoplasmic, R for reticulum, C for calcium, and then A for ATPase. So SERCA. Okay, so I'll colour this in blue here. So the SERCA pump then pumps 2 calcium ions back into the ER lumen. Okay, so 2 calcium ions will go back into the ER lumen, and in exchange it pumps out 3 protons. Okay, and it's an active transport process, so every time it does that, it's going to hydrolyze a molecule of adenosine triphosphate to adenosine diphosphate and inorganic phosphate. Okay, so basically you'll have these SERCA pumps all over the ER membrane, and they will be returning the calcium from the cytoplasm back into the ER membrane, and that will end the signal. Okay, so that's how we terminate this calcium puff, basically. Okay, now I want to talk about the other type of signal, which is this global type of signal here. So we have got the alpha-1 adrenoreceptors being stimulated all over the smooth muscle cell, basically. So everywhere you're getting these alpha-1 receptors stimulated. What does that mean? That means that IP3 is going to go up everywhere, okay? So in the cytoplasm of the cell, IP3 is going up everywhere. That means that along the entire length of the ER, you are going to have IP3 bound to the IP3 receptors. Okay, so if this is the ER, all of the IP3 receptors in the ER membrane are now going to have IP3 bound to them. So they will all be in this state where calcium can actually activate them. And this is going to trigger a calcium signal known as a calcium wave. Now again, the thing which actually initiates the calcium wave isn't known because we don't know where the initial calcium that sets off the first IP3 receptor comes from. We think, and there's a lot of evidence in favour of this, that it comes through channels that are in the plasma membrane, i.e. you let a little bit of calcium in from the extracellular fluid and then that sets off the entire calcium wave. Okay, so we're now going to look at a type of calcium signal known as a calcium wave. Okay, so this calcium that comes in from the extracellular fluid, this will activate one of these IP3 receptors, or a few of them around here. Okay, they will start releasing calcium from the ER. Okay, the calcium will then flow onto the neighbour. Okay, it will stimulate its neighbour because its neighbour, remember, has IP3 bound to it and therefore has these stimulatory calcium binding sites visible. Its neighbour will then release calcium into the cytoplasm. Okay, then the calcium will go onto the neighbour on this side. Okay, so it'll spill over onto here and you'll again release calcium from the ER into the cytoplasm. The calcium will spill over again onto its neighbour. This one will then release calcium from the ER lumen into the cytoplasm, and it will continue on, and basically you will get calcium release propagating along the length of the cell, hence why this is called a calcium wave. Now, basically, just as calcium, once it got to a certain level in the cytoplasm, went from having a stimulatory effect on these IP3 bound IP3 receptors to having an inhibitory effect, the same happens here. So once the calcium has got to a certain level in the cytoplasm, it will start inhibiting the IP3 receptors and they'll start closing. So each one of them will start closing, okay? And then what will happen is the Circa pumps will be working very hard to return the calcium into the ER lumen. Okay, now often these global signals of calcium, often they will be given long term, so it won't just be a short thing, you'll be giving them long term, okay? So for instance, if you have adrenaline being released into the blood, for instance, that will be a long term change. It's not something that happens extremely rapidly. It's not a neurotransmitter that's being terminated extremely quickly. It's going to stay within the blood for a longer period of time, so the cell might be exposed to the stimulus for a longer period of time. So IP3 is going to remain high within the cell for a long period of time. So what will happen is all the calcium will be returned back into the ER through the circuit, and then what actually happens is the wave occurs again. So again, some calcium sets off one of the IP3 receptors and then it propagates all the way along. So what you actually get is repeated calcium waves. So of course this relies not only on the signal being global, but it being sustained, okay? And I've given you the example of if you've got high adrenaline within your blood, then that's going to be a sustained signal basically on the alpha-1 receptors. Okay, now what is known is that these consecutive calcium waves that you can get, the frequency of them is determined by the strength of the signal. Okay, so how many alpha-1 receptors are actually active and producing and resulting in the production of IP3? will determine how frequent the calcium waves is, but the mechanism by which that occurs is just not known, basically. But what's interesting about this is that you've got the intensity of the alpha-1 agonist signal being encoded not by the amplitude of calcium that you're releasing into the cytoplasm. That remains constant. Okay, so the size of each of the calcium waves can't be changed. Instead, what you change is the frequency of them, and that seems very, very similar to what we know happens in neurons. In sensory neurons, so if I just draw a little neuron here, in a sensory neuron, basically the intensity of the stimulus, so for instance if you've got a pressure sensor, the intensity of the stimulus cannot be encoded in the amplitude of the action potential, because an action potential is a digital signal. You either have an action potential or you don't, so you can't vary the intensity of it. Okay, instead what you change is how frequently action potentials are firing down. A more intense pressure on the pressure receptor will trigger it to fire action potentials extremely rapidly, and less pressure will result in less frequent action potentials. So it's just like that. If you douse your smooth muscle cell in an extremely concentrated solution of adrenaline or noradrenaline, it will fire calcium waves across its length at an extremely high frequency, whereas if it's in a very low concentration of adrenaline, then only a few of the alpha-1 receptors will be occupied, okay? You'll have a lower concentration of IP3, and the calcium waves that you get propagating along the cell will be of a lower frequency. But the amplitude of the calcium waves doesn't change because it's a digital thing. You release the calcium, it propagates onto the next, and then you stop releasing the calcium when the calcium concentration in the cytoplasm gets high enough to inhibit the IP3 receptors, basically. Okay, but how IP3 concentration translates into the frequency of the calcium waves isn't known. Okay, so that's a calcium wave, this propagating release of calcium that propagates across the cytoplasm. Now we think that the sort of calcium signal that you get in smooth muscle cells is most likely calcium waves. Okay, so you're getting these waves of release of calcium, and if you sustain the signal, you'll get multiple consecutive waves of calcium. And it's because the Circa pump will be working tirelessly in between the waves to return the calcium from the cytoplasm back into the ER lumen. Okay, so in goes the two calcium ions here. Okay, and then in exchange, it's bringing out 3 protons. 1, 2, 3 there. Okay, right, and that requires ATP, of course. Okay, so that's the 2 types of calcium signal then. Okay, what I now want to discuss is how the calcium being released into the cytoplasm is actually going to lead to the phosphorylation of the regulatory myosin light chain. Okay, so basically, calcium is going to bind to a protein known as calmodulin. So it's time to discuss calmodulin. Okay, so I'm going to give a brief discussion of calmodulin's structure then. So calmodulin has 2 structures depending on whether it's got calcium bound to it or whether it hasn't got calcium bound to it. So we'll start with calmodulin that doesn't have calcium bound to it. And the name for calmodulin when it doesn't have calcium bound to it is apocalmodulin. Okay? And for short, apocalmodulin is usually abbreviated to APO, and then calmodulin is capital C, lowercase a, capital M. Okay, so apocam. Now, apocalmodulin has 2 lobes, okay, the N lobe and the C lobe, okay? And basically, in the unbound state, when you have no calcium bound, it's in this sort of hunched over shape. The 2 lobes are sort of bent back towards one another, basically, and there's this hunch in the structure of the molecule. Okay, now each of the lobes has 2 calcium binding sites on it. So the N lobe has 2 calcium binding sites, and the C lobe also has 2 calcium binding sites. Okay, now basically, 4 calcium ions can come and bind to this apocalmodulin. 1 calcium ion can bind in each of these calcium binding sites. And what happens is when the calcium binds in these calcium binding sites, it causes a change in the conformation of the calmodulin. Okay, so it sort of springs back out, so the 2 lobes move further away from each other. So the whole molecule becomes more straight, and that's in stark contrast to what actually happens to this linker that's between the 2 lobes. The linker between the 2 lobes goes from being a linear polypeptide to being an alpha helix, basically, like so. Okay, so there's the alpha helix. And this is the structure when you have these 2 calcium ions bound in those 2 calcium binding sites on each of the lobes, so overall 4 calcium ions. Okay, and this is what's known as a calcium calmodulin complex. Calcium calmodulin complex. And basically, smooth muscle cells are absolutely stuffed full of calmodulin. Okay, so when the calcium goes up in the cytoplasm of the smooth muscle cell, it will bind to the apocalmodulin proteins, and the apocalmodulin proteins will then convert into calcium calmodulin complexes, and these calcium calmodulin complexes are then going to stimulate the kinase enzyme, which will actually phosphorylate the regulatory myosin light chain. And by the way, the shorthand for a calcium calmodulin complex is to put Ca2 plus the symbol for calcium, and then CAM for calmodulin. Right, so what are calcium calmodulin complexes going to activate? Well, they're going to activate a kinase enzyme, okay, which I'll show here. So I'm going to draw this kinase enzyme in the typical sort of enzyme fashion. So here's the active site. So basically what happens is the calcium calmodulin complex, which you're going to get being formed all over the smooth muscle cell. So we've got this global calcium signal most likely. So you're going to get calcium calmodulin complexes being formed all over the place. Okay? And these calcium calmodulin complexes then bind to this kinase enzyme. Now what is this kinase enzyme? Well, it's myosin light chain kinase. So this is myosin light chain kinase. So it's the kinase enzyme for the regulatory myosin light chain. Okay, and for short, myosin light chain kinase is often abbreviated to MLCK. So calcium calmodulin complexes bind to the myosin light chain kinase enzyme, and this is now going to go off and phosphorylate the regulatory myosin light chain, and once that's phosphorylated, those myosin cross bridges can start binding to the active sites on the actin filaments, and the sliding filament mechanism can begin, and contraction will then ensue. Now, I just want to talk about how you actually relax the smooth muscle cell back down. So once the calcium signal has gone, okay, you will lose the calcium calmodulin complexes, okay, so calcium will go down in the cytoplasm, the calcium will fall out of these binding sites of the calcium calmodulin complexes, and the calmodulin will return to apocalmodulin, hence the myosin light chain will become deactivated, but then you've still got the phosphate groups on the myosin light chains. Okay, so how do you take the phosphate groups off the myosin light chains? Basically, there is another enzyme called myosin light chain phosphatase, okay, which will go around and remove the phosphate groups from the regulatory myosin light chain. So this is MLCP for short, and it stands for myosin, that's the M, light, that's the L, chain, that's the C, and then phosphatase, an enzyme which removes phosphate groups, that's the P. Okay, and if I can just squeeze that in, whoops, no I can't, there we go, myosin light chain phosphatase. Okay, so that then causes the relaxation of these smooth muscle cells. Okay, right, so that then is how our alpha-1 receptors, when stimulated by either adrenaline or noradrenaline, will cause contraction of smooth muscle cells, which is their main localization. Okay, now I just want to discuss a few drugs which act on alpha-1 receptors. Okay, so if I go back to the actual picture of an alpha-1 receptor, here it is. Okay, so we know that agonists for the alpha-1 receptors include adrenaline and noradrenaline, okay? Those are the endogenous agonists. Let me give you an example of an exogenous agonist. So an exogenous agonist for an alpha-1 receptor is phenylephrine. Okay, so phenylephrine is an example of an exogenous agonist for alpha-1 receptors. Okay, an example of an antagonist, okay, so I'll just put that this is an agonist. Okay, an example of an antagonist for alpha-1 receptors is prazosin. Okay, now prazosin and other related drugs, which are antagonists for alpha-1 receptors, are actually used as treatment for hypertension, okay, so too high blood pressure. So the reason that this, it's an antagonist. The reason that this makes sense is because basically all of your blood vessels are surrounded by smooth muscle cells, okay? And these smooth muscle cells, we know, have alpha-1 receptors on, and alpha-1 receptors will cause the contraction of those smooth muscle cells. Now, basically, when the smooth muscle cells contract, the blood vessels constrict, and that's going to force the blood into a smaller volume, okay, and therefore the pressure of the blood is going to go up. So, if we block the alpha-1 receptors, in principle, we should reduce the contraction of the smooth muscle surrounding blood vessels, increasing the volume that the blood is within, and therefore we should decrease blood pressure. Okay, so prazosin is actually used in the treatment of hypertension, and it basically is a drug which will bind to alpha-1 receptors and not stimulate the receptor in any way, okay, but it will stop the agonists such as adrenaline and noradrenaline from being able to bind to that receptor, and hence it will block the activation of the receptor by those other ligands. Okay, right, so that concludes the alpha-1 adrenoreceptor. What we will move on to in the next video is the alpha-2 adrenoreceptor.